In part 2 of protein synthesis, we're going to look at the process of translation. So remember, in transcription, the previous process, we have gone from DNA and we have made RNA, mRNA. Okay, so we've gone from DNA to RNA, and now we're going to go from RNA to protein, and RNA to protein is going to be called translation. And I always remember this, like I said, because it's going from RNA, or nucleic acids, to proteins, which are amino acids. And so it's like it's translating from one language to another. So in this process, we're going to go from our actual code that we wrote down to our final product, which is going to be our protein. So in our analogy of having gone from the recipe in the gigantic cookbook of DNA to our little message that we wrote down as our mRNA, we're going to make our actual food, which is going to be our protein. So in translation, mRNA is going to take this DNA code to the ribosome. tRNAs, those transfer RNAs, are going to bring in amino acids. And then the ribosome is going to put them all together to actually make the protein. So one way to think about this is, let's say that you are in charge of a factory. All right, and you have gotten a message from your superiors that you're going to build this certain product. So we have our materials that we're going to need, and those materials are going to be transported through trucks, and they're going to be transported to our factory, and then we're going to put them together in our factory and make our final product. And that's basically exactly what's happening in this process. The DNA code is going to tell us what to do. We're going to take our materials, which are our amino acids. They're going to be brought to our factory by tRNAs. I mean, just think transport, truck, whatever. tRNA brings those amino acids to our factory, which is our ribosome, and we're going to put them together to make that protein. This is more what it actually looks like when we're talking about protein synthesis. This looks like the picture in the back of the room. So in this picture, we have our mRNA, so that serves as our code. Here's our ribosome, our factory where everything's happening. We've got our amino acids that are being put together to make our protein. Our tRNAs, notice how they literally look like a T. And they are bringing the amino acids to be put together to make our protein, which is our final product. Now, when the proteins are put together, the ribosome is actually going to make a certain type of bond between the amino acids called a peptide bond. And this is going to form a polypeptide chain, which is another name for a protein. It just means many peptides. So for example, we have an amino acid, and then another amino acid is going to be bonded to it with a peptide bond, and so on to form our polypeptide, which is our protein. Now let's look at how this code works. So if you remember, we have codons, and those are 3 letters, 3 bases that form sort of the words that create the code for our protein. Each one of those codons is going to code for a specific amino acid. That's really important to remember. Each codon codes for an amino acid. And it's the order of the amino acids that determines the type of protein. All right, so the order of the codons determines the order of the amino acids, which determines the type of protein, which determines the trait. So this looks like a pretty crazy chart, and basically what this is this is a chart that you're going to be using that shows you how to link mRNA codons to their amino acid. So we're going to, basically I'm going to show you how this works, but all this does is help you determine if I have this particular mRNA codon, what amino acid does it code for? So let's see how this works. So here's my mRNA sequence. All right, so let's start with AUG. AUG is going to code for a specific amino acid. So what I'm going to do is I'm going to look at the first base of my codon, which is an A. And what this chart tells me is I'm just going to look on this row. The second base is a U, so I'm only going to look in this column. So this box has the correct answer in it. And then I'm going to use the third base, or the G, to see that AUG codes for methionine, which is an amino acid. And it's also the start codon, which basically tells all the ribosome's machinery and everything to get started. And most of the time what we're going to do is we're just going to abbreviate these things with the first three letters. So MET for methionine. You don't need to memorize any of these amino acids. All right, GCC is going to code for alanine, UCA serine, and UGA is a stop codon. So notice that UGA, UAA, and UAG are all stop codons. These tell the ribosome, stop, everything's done. All right, so how does this process work? Here we have our mRNA. It's going to attach to the ribosome. And notice the ribosome's kind of got these rooms. This is going to be where the action happens. I have a tRNA. And notice that this tRNA has bases at the bottom of it, and those bases are complementary to those that are on the mRNA. So the first mRNA codon is AUG, and this tRNA has UAC. That's what allows it to bring the right amino acid to match up with the correct codon in the mRNA. So this tRNA is going to attach into our ribosome, bringing its amino acid, and then the ribosome is going to shift down, just like a assembly line. And then the next tRNA that matches up with the right codon and brings its own amino acid is going to come in. The ribosome is going to shift down, and notice the ribosome has now put those two amino acids together to start forming our protein. All right, the next tRNA comes in, we shift down, that first tRNA goes away, and then in comes our stop codon, because this is a pretty short protein. So when the ribosome shifts down, there's nothing there to bond to. Tells the ribosome, we're done. Everything kind of breaks up and goes off on its own. And now we have left our protein. So how does this whole process of protein synthesis go together? Well, remember we start with our DNA code, and through transcription, the process of transcription, we are going to transcribe that code into mRNA. And then through the process of translation, we're going to translate that mRNA code into amino acids, which form our protein. So if we look at this, like you're going to have to be able to do in some problems, here I have a DNA sequence. Now, if I tell you to transcribe this DNA sequence, you're going to create the complementary mRNA sequence. And then notice I have identified the codons, because that's going to be the next step. When you translate them, you're going to take each one of those codons and you're going to find the amino acid that it goes with on the codon chart. And you're going to find the order of the amino acids, which in this case would be histidine, threonine, proline, methionine, and tyrosine. So remember, protein synthesis, DNA to RNA to protein, DNA to RNA transcription, RNA to protein translation.